Good afternoon, my name is Jefferson Susoni and I would like to talk to you today about anxiety. As we are aware, Drosophila Melanogaster is a model organism whose genome is 60% homologous with that of humans. Another parallel between the fruit flies and humans is that 75% of genes responsible for human disease have homologs in flies, thus making the organism a perfect candidate for research. The ADAA estimates that 6.8 million adults suffer from generalized anxiety, with only 40% of cases receiving treatment. The mean total medical cost for individuals diagnosed with anxiety disorder comes to a whopping $6,475 per year, and a lot of this cost is due to the expense of the medicine. So research into alternative medicine for individuals with anxiety would be number one, of great use, and number two, it would greatly impact the lives of these individuals by reducing the cost of their living expenses. Darwin proposed that all animals have emotions. He wrote this in the book Emotions in Humans and Animals. Emotions are described by science as a natural phenomena that occur unconsciously and affect the body's physiology. Feelings, on the other hand, are more subjective, and science doesn't really do well with feelings. Arnold M.B. states that emotions occur in three stages. Number one is perception, number two is the actual emotion and how the emotion is affecting your physiology, and number three is the expression of that behavior. We cannot study the fly's perception because we cannot communicate with it. We cannot ask it what it thought when it felt isolated, right? We can, however, look at the behavior of the fly to try to deduce what its emotion was while it was performing the behavior. Now, how do we do it? How do we know that a fly is anxious, right? So a really cool little feature that flies have is that they tend to walk the perimeters of arenas when they are trapped, meaning that if you trap them in a cube, they will constantly walk around the walls. The cool thing is that they display something called central phobism, which means that they are afraid of the center, meaning that they move on the perimeter of the wall and rarely move to the center. They will sometimes cross but never dwell the way that they dwell around the perimeter of the arenas. This is really cool. So research has already been made into the genes that play a role in anxiety, and science is working to define the specific genes that play these roles. In an enclosed arena, flies, individual flies, tend to stay right to the walls during spontaneous locomotion, like I stated before. A behavior proposed to be tied to anxiety, and they coined it as WAFO, WAFO. In the publication Ancient Anxiety Pathways, Drosophila Defense Behaviors, researchers manipulated the GABA receptors in flies in order to affect central phobism. Like I stated before, central phobism is basically the desire of the fly to move away from the center of a surface. WAFO then was calculated by tracking the data for 10 minutes. Each experiment was performed for 10 minutes on individual flies. Each fly was observed individually. The fly was allowed to walk the arena, and the pattern of movement was tracked. This pattern, surprisingly, is also really familiar. It's really close to the pattern of rodents. And what researchers did is that they cross-referenced the patterns with 3 essays that were performed on rodents in which the rodent displayed a similar behavior. And they deduced that that behavior was also anxious. So we have 2 animals that are displaying similar behaviors when they are anxious, right? So that's cool. Researchers then calculated the mean distance from the center, right, of the enclosed arena to each fly. And that is what they deduced to be like the mean. So first they calculated the distance of the pattern walked, and then they grabbed each observation of the flies and created a mean distance, and that mean distance was basically the main number that they compared like the effect of WAFO and how it was affected by the dose of diazepam. So what is diazepam? I'll explain that in a second. That makes no sense. Science is working to to define the specific genes that play a role in anxiety and other related issues. In an enclosed arena, flies stay close to the wall during spontaneous locomotion, a behavior proposed to be the expression of anxiety in flies and coined as wakeful. In the publication, Ancient Anxiety Pathways Influence Drosophila Defense Behavior, researchers manipulated the GABA receptors in flies to affect centrophobism. As I stated before, centrophobism is the desire of a fly to move away from the center of the surface that they are currently walking. Meaning that when a fly walks a surface, let's say that this is the upper right corner of the cube, and now we're looking at the wall. The fly will walk the perimeter of the wall, staying close to the other 90-degree angles, right there. So it will walk around the perimeter and avoid the center, and that is what is called as centrophobism. Wakeful was then calculated by tracking the data for 10 minutes. Each fly could walk the arena and the pattern of movement was tracked. The cool thing about this pattern is that it's really close to the pattern that mice display. Scientists basically cross-reference the pattern from the fly, and they cross-reference to 3 assays on rodents, and they notice that they have similar behavior, similar patterns. They deduce on the rodent assays that this behavior is tied to anxiety, and that being the case of one animal gives validity to the hypothesis that flies walk around when they are anxious and they avoid the center. Cool. Then what scientists did is that they had a control experiment, like a control group that was not fed a active ingredient called diazepam. It's a anxiety it's an anxiety like medicine. So a group was not given diazepam, a group was given diazepam. They were observed for 10 minutes and allowed to walk the arena. What researchers found was that the group that was given diazepam showed decreased wakeful, meaning that they would venture into the center a little more and stay in the center for a little longer periods of time before moving on to walking the perimeters again. Another important fact to add is that the amount of diazepam was tripled. So the experiments were repeated, and as they were repeated, the dosage was increased. And when the dose reached 3, when it reached a triple dose, is that they actually notice a significant change in wakeful. So that is important. Now, very little research has been made on the effects of THC on anxious behavior, or very little research really on THC. Now, I propose creating an assay in which THC concentrates are used to affect wakeful. Well, they're used to affect wakeful. We do not know whether it will be by increasing it or decreasing it, and that is what I want to gather, that information that I want to gather. What I want to also do is basically create an experiment that is similar to the experiment in which the flies were given diazepam. I actually want to repeat the same assay as a measurement, like a standard, and compare THC's effect to that standard. So I want to have 3 groups. I want to choose a total of 15 male flies. Each one of the flies will be male, right? Wild type, and they will be raised in a social setting. After being raised in a social setting, they will be isolated individually and separated into 3 groups of 5. Group 1 will be given THC. Group 2 will be given diazepam, and group 3 will be given only food. Also, it's important to add that each group will get food with a dosage of either THC or diazepam, and the third group will not have anything other than their food, only their food. The flies will be individual flies will be observed for 10 minutes. So we have group 1 being observed for 10 minutes, total of 5, separated, right? Each one is on their own little vial, and then group 2 another 5, group 3 is another 5. Everybody gets observed for 10 minutes. We will repeat the experiments 3 times, and each time the dosage of THC will be doubled, will be added at 10%. So I will first start with 10%, add 20 on the second observation, add 30 on the second observation. Same thing with diazepam, start with 10%, at 20, at 30. Again, 10 minutes and only male flies. After observations are completed, I hypothesize that THC will have similar effects to diazepam. The control group will surely have a higher amount of WAFO than group 2, because group 2 will be given diazepam, and we know for a fact that diazepam decreases WAFO. So it is of course that group 3, the control group, will have a higher WAFO, and group 2, the group that was given diazepam, will have lower. What I want to do with THC is observe. My hypothesis is that it will be similar to diazepam, and therefore more research could be done into THC as a alternative method to treat anxiety. So that is my talk, my line and talk for the idea that I have in regards to the essay for THC and its impact of anxiety in flies. Hopefully down the line this could be improved upon, and maybe we can see how is it that THC also affects anxiety in social settings. But before that can occur, I feel that we first need to nail down the effect of anxiety of THC on an individual that is not prompt to being anxious, an individual that is just normal, and see how it goes. My name is Jefferson Sasonia. It was a pleasure to talk to you, and this is my little talk. Take care of yourself.